So the last time we got together, we were talking about gene expression, and we started with the first part of gene expression, which is transcription. And today we're going to move on to the second part, which is translation. So one thing to just think about real quickly is when we talk about transcription, you'll notice the word script in the middle of that. It's as if the DNA is writing out an order for the protein that it wants, and it's giving it to the messenger RNA. And now we're going on to the translation part where we're going to take that message and we're going to put it, we're going to translate it into an actual protein. So you saw this slide last time, and I just wanted to quickly review it so we're all on the same page. And our overview of making proteins is we're starting with our DNA in the nucleus, and we have our template strand and our complementary strand. And the messenger RNA then is going to be created from our template strand. And note that the messenger RNA, every 3 nucleotides equals a codon. And codon is just what it sounds like. It is a code for a particular amino acid. And so the messenger RNA is going to be transported from the nucleus through the cytoplasm into the ribosome, which is where our proteins are made, and that's where translation occurs. And so we're going to start with that today. So the translation portion is when we take the messenger RNA and from it make a protein. So this is taking place in the cytoplasm with the ribosomes. So ribosomes are in our cytoplasm, and it involves our second type of RNA, which is tRNA, and which stands for transfer RNA. So far, our messenger RNA has basically looked like a straight line that was complementary to the DNA. But RNA doesn't always have to be in a straight line. It can be folded and have a U shape. And in fact, that is what tRNA looks like. So this is an example of tRNA. So even though it's called transfer RNA for the function that it provides, it's also helpful because it looks like a T in its structure. And so it has several different portions to it. So let's look at that real quickly. We have an amino acid at the end here, usually leucine, and through hydrogen bonding, it will complement with itself to do some folding like this, right here and right here, and then also down here. And then it has a portion called an anti-codon end. And that's something that should be somewhat self-explanatory. We've talked about codons in our mRNA that is a series of 3 nucleic acids. And so the anti-codon is also a series of 3, but they are complementary to our codon. So each transfer RNA recognizes one mRNA codon, and it recognizes it by using the anticodon, which is that complementary sequence. So the tRNAs are out there in the cytoplasm. They recognize the mRNA with their anticodon, and they carry the amino acid indicated by that messenger RNA codon to that mRNA site. So when we look at the bases that we talked about, we have adenine, guanine, cytosine, and uracil, right, for all our RNAs. So every possible combination of 3 that we use those for gives us 64 different codons. And for those 64 different codons, there is one tRNA for every possible mRNA codon. So they're very specific. Okay, so if these tRNAs are in the cytoplasm and they have to bring an amino acid over to the messenger RNA, how do we know, or maybe I should not how do we know, what makes sure that the tRNA is attached to the correct amino acid? Well, that is the role of what we call amino acyl tRNA synthetase. I know that's a mouthful, but it's an enzyme that makes sure that the amino acid is correctly placed with the right tRNA. And that is a process that involves ATP, as you might expect. So it basically makes sure that we have excuse me, an amino acid coming in, and of course as an enzyme it has a particular binding site. ATP does as well, right? And then we have the tRNA that comes in and binds, and so then it attaches the amino acid to the tRNA and then it's released from the enzyme. And so that is how every amino acid is attached to the correct tRNA, and that enzyme is called amino acyl tRNA synthetase. Okay, so here's our overview again on making proteins. We've done transcription in the nucleus. We've come up with the mRNA that has been corrected and sent out into the cytoplasm, and then we are now looking at translation in the ribosomes to make proteins. Okay, so we said there's at least one tRNA for every amino acid. So you can picture that as our codon on our mRNA with our 3 nucleic acids, the anticodons on the tRNA, and then here we have the amino acid that's been attached. And so as we go down the mRNA, we go to each codon and have the tRNA attached. When the tRNA is full of its amino acid, we call that a charged tRNA. So when it brings in the amino acid, the charged tRNA binds the codon on the mRNA. So here we are just looking at it overall. Our transcription, we had our template strand of our DNA, and then our messenger RNA that was created in the 5' to 3' direction. And from that we have our anticodons that come in and bind to the codons in the messenger RNA, remembering that the start codon always is coded for methionine right here. Okay, so these are charged tRNAs that are binding to our codons on our messenger RNA. So this is what it looks like, because this is happening on our ribosome. So our ribosome, we have the mRNA right here in the small subunit, and then our transfer RNA comes in on the large subunit, and our peptide chain is created. Now, let's take a look at our ribosomes, and because it has different sites that I want you to know about. Okay, so like we said, the ribosome comes in a large subunit and a small subunit. So let's start with our A site. Our A site on the large subunit is where the tRNA first arrives. So it first comes in, so you can use arrives for the A site. The P site is where the tRNA is holding the polypeptide, or the amino acid chain. And then the E is the exit spot for the tRNA. Okay, and this, of course, down here is where our messenger RNA is going to be. Hopefully that makes sense to you. So there are 3 steps of translation, just like there were 3 steps of transcription, and amazingly they are the exact same. So initiation, elongation, and termination. So in initiation, that's when the first tRNA carrying the methionine is called, and that's called the initiator tRNA. Because remember, that's where the start codon is. So here's our, let me get my pen. Our start codon, AUG, which codes for methionine, so that begins our translation. So it's initiated right there. Our second step is elongation, and just like you would expect, we have multiple tRNAs coming in, and we can have 2 tRNAs at one time on our ribosome, so they can both be acting at the same time. And so our amino acids come into the A site, and And then they're going to, of course, leave the exit site. In the meantime, we have our amino acid chain beginning to grow. And that's our P site, remember, for polypeptide. Our step 3, of course, is going to be our termination. So a protein comes in to make sure that it's terminated, and that's called a release factor. And that release factor recognizes one of the 3 stop codons, and it stops that translation. So when it does that, then we have a release of our polypeptide, which is, remember, our primary sequence of our protein. Okay, so let's say that this is the polypeptide that we are creating. So if we take a look at this, we've started with our start codon, of course, which is going to put in our methionine amino acid. And then we have all these other amino acids that are then ligated together to form our polypeptide. And oftentimes one or a few of these amino acids are used as a signal to send our polypeptide from the ribosome on to the next area where it's going to get processed. So this is going to look familiar based on what we've talked about before with the different organelles and what they do. So here is our rough endoplasmic reticulum. This is the primary place for protein synthesis. It can also happen in free ribosomes within the cytoplasm. But there are so many ribosomes on the rough ER, that's where most of our protein synthesis occurs. When it is completed, oftentimes we have transport vesicles that then take those proteins to the Golgi apparatus. And remember, one of the functions of the Golgi apparatus is to modify proteins from the endoplasmic reticulum. So that's where we start all the folding processes, and all those additional enzymes that make sure that the proteins get folded properly are in the Golgi apparatus. Once they're all folded properly, if the protein is going to be used outside of the cell and in a different location, then it is then packaged in one of the secretory vesicles, and then through excytosis it is sent outside of the cell. And that is the destination of our protein that started with our DNA and transcription through translation, modification in the Golgi apparatus, and sent out. So here is the summary of gene expressions in eukaryotes. So we start with our DNA in the nucleus, and that serves as a template for our messenger RNA. So the messenger RNA is created, and then before leaving the nucleus, remember it goes through those 3 steps of being processed, then it leaves out the nuclear pore into the cytoplasm, and either in free ribosomes within the cytoplasm or ribosomes on the rough endoplasmic reticulum is where translation occurs. And so that means that the tRNAs with their anticodons are brought in to match the codons of the messenger RNA, and when they are charged tRNAs, they bring with them their amino acids that can then build the polypeptide chain. And so polypeptides are synthesized, and then they are sent on to the Golgi apparatus where they are modified, folded correctly, and then they can either be used within the cell or sent out through exocytosis. So the major differences between prokaryotic versus eukaryotic gene expression is that in prokaryotic cells, we have transcription of the DNA straight into RNA. There's no nucleus, so we don't have to worry about going through a nuclear pore or anything like that. The mRNA is made, there's no pre-mRNA, it's just straight mRNA because there's no modification to it. So once the mRNA is made, it's directly translated into a polypeptide by a ribosome. Our eukaryotic cell, however, transcription is still taking place, but it's taking place within a nucleus. We make basically our rough mRNA or pre-mRNA, which is then processed into mRNA, and then it is released through our nuclear pore into the cytosol where it's translated at the ribosomes. So when we look at it in comparison, gene expression in prokaryotes happens entirely in one place, whereas in eukaryotes, the transcription happens in the nucleus with translation happening in the cytoplasm. In the prokaryotes, the RNA is not processed, whereas it is in our eukaryotes. And in prokaryotes, transcription and translation occur at the same time. I didn't mention that before, sorry. So because it's all in one location, it all happens on what we call polyribosomes. So transcription and translation happening at the same time. In eukaryotes, the transcription and translation are separate events in separate locations. So here is an image of that polyribosome that we were talking about. So in this image, we have our DNA right here. So you can actually see it in the micrograph right here. There's the RNA polymerase. And then as the mRNA is being produced, the ribosomes then attach to the mRNA, forming this polyribosome unit, and then proteins are produced right there, all happening in one place at the same time. Okay, so let's talk about if we have problems within our DNA that aren't corrected. So the top left here represents what we would expect with our DNA. The complement is correct. So we talked about before that we could either have deletions, additions, or base substitutions that could be problems. So how does this play out through gene expression? So each of those is called a point mutation. That means that there's one change in a nucleotide sequence. And that seems like such a small thing, but it actually turns out to have pretty large consequences, which that in itself ought to blow your mind for how many times we have gene expression and that it's done correctly. That by itself should set you in awe. Okay, so deletions, additions, and base substitutions are all examples of what we call point mutations, one change in a nucleotide sequence. So in the next slides, I'm going to show you what can happen with a point mutation. And specifically, we're going to look at the disease called sickle cell anemia. Okay, so our normal hemoglobin, so that hemoglobin is that protein in the center of your red blood cell that allows you to carry oxygen to all parts of your body. So as we go along in our DNA, and we have CTT as one of our codons, and then here are our anticodons, GAA, which codes for glutamine. What happens in our point mutation, let's say that this one here in the middle gets thrown off, and instead of CTT, we now have CAT. Well, that changes the anticodon then to GUA. That GUA codes for a totally different amino acid, valine. And the result is a sickle-shaped red blood cell because we have changed the hemoglobin protein. So what we call normal DNA, the other term for it is what is called wild type. We have wild type versus mutant hemoglobin. So here we go with our wild type DNA at the top, with the mRNA being processed just the way it should be. And then here is our mutant hemoglobin with our substitution right here of A, and that provides a valine as opposed to a glutamine. And this is actually a micrograph of what your red blood cell looks like. It's not nearly as functional. So the interesting thing is that if we have a base substitution at the DNA level, at the protein level that can result in 3 different types of mutation. So when there's no mutation, for instance, again we have our DNA TTC, mRNA level complementary would be AAG, and that results in a lysine amino acid. So with these, if we have a point mutation on the end and we substitute the cytosine for thymine, our mRNA level is going to be an adenine instead of a guanine. So interestingly enough, AAG and AAA both code for lysine. So this is what we call a silent mutation. So you wouldn't notice a difference on that one because it still codes for the same amino acid. Now, if we had a point mutation at the beginning of this codon and we replaced the thymine with the adenine, our mRNA would be a uracil at that point then, and the codon would read UAG, and that would mean a stop sequence. So our protein would stop being made at that point, in which case we really just have a part of a protein and not a full protein. So that's what we call a nonsense point mutation. A missense then is this next point mutation, and let's say we substitute our thymine for a cytosine. That would result in a guanine in that point for the messenger RNA, and AAG codes for arginine. Well, that will mean the wrong amino acid. Additionally, we could have replaced it with a guanine. The mRNA level would have been a cytosine, and then we're coding for yet another amino acid. So that's the wrong amino acid in that particular sequence, which may at the primary structure not quite look bad, but by the time you start getting into secondary structure and tertiary structure, all of these chemical bonds make a difference in how the protein is folded, and so we're going to have the wrong protein. So the way we can look at this is with the DNA substitution, we have a silent mutation where the nucleotide change doesn't affect what amino acid is coded for, a nonsense mutation where the nucleotide change codes for a stop codon and chops the amino acid sequence, or we can have a missense mutation where the nucleotide does code for the wrong amino acid, completely messing up the protein. So hopefully that explains then how with sickle cell, when we have a point mutation, it codes for the wrong amino acid, and therefore the hemoglobin protein does not fold correctly. So this is a missense mutation. A fourth type of mutation is at the DNA level is when you have either an addition or a deletion. And at the protein level, this can result in what we call a frame shift mutation. So if our wild type or normal sequence of DNA looks like the frame on the top, and it gets read, if you think about this as a codon, each frame being a codon, and it would be read just like that, then if we add or delete a particular nucleotide, then that throws off the codon. So now we would be reading across like this, right? And so that is called a frame shift mutation. So this is what it looks like if we're looking at our DNA, our normal or wild type DNA. Here we have our codon and our anticodon. So for here, we are making a protein that has lutein, alanine, cysteine, but when we have our frame shift, we've added a guanine in here. And so whereas we're normal here until we get to this addition, now it shifts and we have arginine, methionine, and that's not what this was supposed to be coded for. So that's called a frame shift mutation. So it's an addition or a deletion of a nucleotide that changes the order of threes. Okay, so that is your transcription and translation for gene expression, and then how that can go awry. Okay, so that is it for our gene expression. Make sure you review that and you know again all the players and what can go wrong.